hide it. Maybe like this. It's better.
Now it's in.
Okay, like this.
Yeah,
That's perfect.
Okay, yeah.
that's really good.
I don't touch anything more.
All right, should we start? I'm going to do a short introduction of your work, and then you can start.
Okay.
So, Dr. Jessica Dubois is a researcher at INSERM, specializing in early human brain development. She earned an engineering degree in physics from Ecole Centrale de Paris in 2001, followed by a master in interface between physics and biology in 2002, and a PhD in physics in 2006. I didn't know your PhD was in physics. I was like super impressed. From University Paris Sud. Postdoctoral work included position at Geneva University Hospitals and the UMR 663 in Paris. Since 2019, she has been part of the Neurodiderot unit in Paris, and she's co-leading the in-depth team. Is that right? Still up to date? Yeah. So today she will present us her latest research gathered under the exciting title, Brain Development in the First 1,000 Days. Thank you very much, Jessica.
Thank you, Michel, for the introduction and for the invitation to present this work. So I'm very excited to show you my work, but also other things, more general things on brain development, as I thought that maybe that would be interesting for you. So as you might know, all the amazing behavioral acquisitions that the baby has after birth is relying on intense development of his brain. And this brain development is actually particular in the human brain because it's very intense during pregnancy, but also in the first months after birth. So this makes the environment to play a role in this development. If you compare to non-human primates, actually, the intense phase of development is mostly focused during pregnancy rather than after birth. So this intense brain development is actually relying on many mechanisms. So you have these complex plots on the right side detailing all the mechanisms involved in development. So it's cell proliferation, neuronal migration, etc. So, but this plot is actually to show you this diversity of mechanism and also the fact that they are overlapping over time and potentially interacting. And these mechanisms are depending on genetic, epigenetic factors, and also environmental factors, as I mentioned before. An important thing also about development is that close to birth, preterm and after birth, it's a very intense period of neuroplasticity, meaning that everything that happens to the kid and potentially interventions might have long-term effects on the development. So in my work, I'm particularly interested in this red box that I show here. So here it's actually birth here, and you have the last weeks of gestation and the first months after birth. And I'm trying to get some markers with neuroimaging to get insights on this mechanism. So with neuroimaging, actually, we can't really have access to neuronal migration, for example, but we have some proxies, some markers with neuroimaging and with techniques in particular like magnetic resonance imaging, MRI. And we can also have access to the functional activity of the brain through electroencephalography, EEG. But actually, you can imagine, I guess, that using these methods in babies is quite challenging. So typically in MRI, what we try to do is to have the baby asleep during the MRI acquisition so that the baby doesn't move too much and so that the MR images are not artifacted by motion artifacts. So here are how we install these little babies with MRI and with EEG. Another important aspect that I would like to tell you in this introduction is actually the fact that prematurity and disturbances that happen very early on during gestation might have a great impact on this developmental mechanism, completely changing possibly the trajectory of development, and this might have some impact on the whole outcome of the kid, the way he will develop his acquisition, his learning, etc. during childhood. So potentially, I hope I can cover these different things in this talk. So I would like to show you a few basics about early brain development and how we can have some markers with MRI of this maturation. Then I will try to give you some examples of anatomofunctional correlates in infants, and then mention some perspectives on the studies that we can lead, that we can have on the impact of prematurity, and also for the prediction of outcome in babies that are at risk of developing disorders like ASD or others. So let's start with the few basics. So you had a great talk about brain development a few weeks ago by Andreas Jacob. So I will be quite quick on this. So you probably know that the neurons are formed in zones that we call proliferative zones that are central to the brain. Then the neurons migrate to the periphery to form the cortex. And there are two kinds of migration, the radial migration and the tangential migration. And here you have actually some slices of the brain showing these proliferative zones in the middle, what will become the cortex here, the cortical plate. And in between, you have actually two zones during pregnancy. So here it's the delimitation in a term baby, so at birth. You have these two zones, which are the intermediate zone that will become the white matter, and also the subplate, which is composed of neurons that are the first to migrate and that will form some connections with other parts of the brain. So if we try to have access to these structures, tissues in development, we can use MRI. So here you have examples of T1 weighted images and T2 weighted images. On the left, it's a preterm baby at around 7 months of pregnancy. And here you have an adult. So you probably recognize that these are brains, but there are several differences. In particular, the contrast of these T1 weighted and T2 weighted images are quite different with development. And this is because the relaxation times are changing, mostly in the white matter, more than in the gray matter. And this is due to this mechanism of development that we will see after. So actually, during the first year, you have an inversion of the contrast in T1 weighted and T2 weighted images. If you look at the brains macroscopically, one amazing fact is the brain growth during the last weeks of pregnancy and also the folding process. So the fact that the brain has these little folds that are formed. So here it's what we get from MRI by using post-processing tools to get the cortical surfaces. So here it's a preterm at 7 months of gestation compared to a newborn at term, just born, and an infant around 3 months of age. So you have a big increase in brain size. You can see this when you look at changes with age. Here you have an increase in cortical surface. And if you look at the changes in the folding index, actually you see also an increase that is very intense during the last weeks of pregnancy, but then you have a kind of plateau, like a deceleration of the folding process. One interesting way to get to look at these folds is to look at folding patterns with objects that we call sulci. So here it's a framework developed by the team of Jean-François Mangin in Neurospin, in which actually he identifies sulci objects, and from these sulci we can identify different measures, and I will go a bit further into this in some of the next slides. So here you have examples again in preterms at 7 months of gestation, at term equivalent age, so at the age at which the baby was supposed to be born, compared to an adult. So you already see that at term equivalent age, you have complex geoification with all these sulci that are already in place, but they get more and more complicated with development. Another important aspect of development is changes in cortical microstructure. So within the cortex, the neurons develop connections with the surrounding ones. And you can see here at 36 weeks of gestation, you have mainly a few connections and these apical dendrites that are here. And with development at birth, 3 months, 6 months, you see that connections are developing in all directions. So it's what we call exuberant dendritic arborization, exuberant synaptogenesis. Then in a second step, actually, the connections that are not used, that are not relevant, are suppressed, they are pruned, and this comes later on. You have this pruning process that comes by 4 years, 6 years of age. So you see that here the microstructure is less complex than at the time of 6 months, 2 years of age. This synaptogenesis, it happens actually at different rates across brain regions, meaning that for different functional networks that get mature at different times, there will be different rates of synaptogenesis. Typically, in red here you have the changes for the visual cortices compared to prefrontal cortices here in green, and these prefrontal cortices are actually maturing much later and much slower than sensory regions like the visual ones. One interesting technique to look at the microstructure is, of course, diffusion MRI. So I won't explain you how diffusion MRI works, but I wanted to show you some maps that have been estimated through the model of diffusion tensor imaging. So again, on the left, you have a preterm, on the right, an adult. And I hope that you can see that what is changing a lot is the values of diffusivities, because initially the brain contains a lot of water, and also the anisotropy is increasing a lot with the myelination process that we will see later on. So with diffusion MRI, we can actually have a look at the cortical microstructure and this process of high dendritic arborization that I've mentioned before. And different groups have actually described that very early on in the preterm period, you have actually a simple microstructure which leads to a strong anisotropy in the cortex. So this is in the preterm period, typically here at 7 months of gestation. But by the age of term equivalent age, actually the connections have developed in the cortex in all directions, and this decreases the anisotropy. So when you look at the neonatal image in a full-term baby, you don't see anymore this high anisotropy in the cortex. So we have been able to measure this in preterms that were scanned twice during development, once early on, once at term equivalent age. And here you see a strong decrease in the anisotropy within the cortex. During infancy, we can measure other metrics like axial diffusivity, and we also see differences with these differences that are different across brain regions. Typically here in the central cortex, you might see that the slope of the decrease is less intense than in the frontal cortex. Now, if we focus on white matter connections, we can have a look at when and how they develop in the baby brain. So the development of long-distance connections actually take place mostly during gestation. This involves actually subcortical structures like the thalamus and also these transitory structures that I mentioned, the subplate, and this starts very early on, from 12 weeks of gestation. By term equivalent age, actually, all the long distance fibers are already in place. So you have actually this nice summary by the team of Manon Benders showing when the fibers start growing for projection, commissural, and associative fibers, and when these fibers are supposed to be fully developed in babies. So for these long distance connections, at term, they are developed, but potentially they have been too much developed. So there is also this process of pruning that will happen for these long distance fibers after birth. And regarding the development of short cortical fibers, actually this process is supposed to take place mostly after birth, but this is not completely clear. The timeline is not well known, actually, but possibly during the first year after birth. We can have a look at these connections using, again, diffusion MRI and tractography. So here you have some examples of fascicles obtained in infants. There have been some nice work from the team of Stam Sotiropoulos to develop some atlases to get reconstructions of the main fibers. And so one interesting aspect that I would like in the future to be able to explore is actually this pruning of the fibers that I mentioned before, and that is supposed to depend highly on activity. So here, for example, you have the corticospinal tract that was reconstructed. So it's a coronal view. And actually, what we know from other studies, not relying on diffusion MRI, is that initially this corticospinal tract contains both contralateral and ipsilateral connections, meaning that, for example, the left tract is connecting to the right hand, but also initially to the left hand. But with development and with good activity, let's say, of the brain and of the motor limbs, actually you have a pruning of the ipsilateral connections so that only the contralateral connections are functioning. But what is happening when there is an early brain injury is a reorganization of this connection, or it's not really a reorganization, but the fibers that were supposed to be pruned don't prune, so that, for example, for this left tract here, it's still connected later on during childhood with both the left and the right hands. So far, we can't have access to this process of pruning with diffusion MRI, but I think it's an interesting aspect that should be developed in the next years. Besides, the white matter is myelinating. So you probably all know the role of myelination of axonal fibers to increase the conduction speed of neural information, but also to stabilize the functional networks because the fibers won't prune if they are myelinated. This process, you might see that here on postmortem images, that it doesn't occur at the same rate in all the brain regions. So initially it starts in central regions and then it's spread to the periphery. This again, it's something that we can measure with diffusion MRI with different metrics such as radial diffusivity when we use diffusion tensor imaging. And we can also measure the myelin related water through techniques that are based on relaxometry. So here, for example, you have maps of water related to myelin. So it's a fraction of this water. And the higher the values, the darker the regions are. And so I hope you see that this myelination is, we observe that it proceeds from the central regions to the corpus callosum, to the occipital regions here, and to the whole periphery of the brain. So that's what we can do actually with MRI to look at markers of anatomical maturation. And now I would like to give you a few examples of how these markers of anatomical maturation relates to the functional development of babies. So maybe before that, I can mention some of the amazing behavioral acquisitions of babies after birth. So it goes in the same direction of what you were mentioning at the beginning. So the little baby actually already does a lot of things. So its memory is already functioning quite well. During pregnancy, it can remember things, and we can test this after birth. Very early on, neonates have some strong discrimination capacities. So these have been observed both for language, typically for phonemes, and also for the visual processing of faces. But with development, actually, the baby will lose some of his discrimination capacities and improve the ones that are really relevant according to its environment. So he is improving actually her expertise according to the environment. So that's why actually the role of the surrounding persons is really important. And we talk about sensitive periods, which means periods that are really important to develop a functioning brain. And with this environment, actually, the role of social interactions is actually really important. besides of this, of course, the baby starts to have multimodal perceptions after birth, and he starts actually quite early on to integrate of his stimulation. And what he's doing is what we call the brain is learning through statistical learning. So it has already early on some models, and then confronting the model to the stimulation and changing the model if errors were made initially. So how do all these possibilities and acquisitions relate to the developing brain? It's actually something that is not well known yet. And what we others and Howard have found is that very early on, there is some functional specialization already, but this specialization is still quite incomplete. So in the next slides, I would like to relate the markers, the anatomical markers that I mentioned before, with this functional development that we can assess. So I will start first with the investigation of cortical microstructure and myelination that show an maturation of brain networks that is asynchronous, meaning that it doesn't happen at the same rate and with the same calendar across brain regions. So this cortical microstructure is actually different across cortices, also in adults, is something that is known. And the myelination, as I said, it's progressing first in central regions. And if you look at subcortical white matter, you see that mostly it's regions related to sensory functions and motricity that are myelinating first. So here it's the famous calendar by Yakovlev and Lecour showing the progression of myelination. So you see this high variability in the starting points and also in the duration of the myelination across the regions. So through the markers that we have with MRI, we can have access to these maturational differences across cortical regions, across white matter bundles. And what is interesting with MRI is that we can quantify these differences and potentially follow them during development if we have longitudinal MRI. So it's the big advantage of this. And of course, we can relate this to behavioral acquisitions. And so the main point here is that at the whole brain level, we have an early maturation of brain networks that are involved in sensory perceptions and motricity compared to associative regions. But the link with anatomical features is not well known between these anatomical features and functional specialization. And so I will start first by a recent study from my former doctoral student, Andrea Gondova, in which actually she tried to compare some microstructural markers and functional connectivities that we assess with resting state fMRI. So for functional connectivity, it's quite classic, nothing new. But the original point of this study was to look at what we call the microstructural connectivity, meaning that we looked at the connectivity of different gray matter regions, cortex and subcortical regions, and we assessed the correlations in the microstructural properties across regions to get a marker of microstructural connectivity. So first, she assessed whether there is some relationship between this microstructural connectivity and functional connectivity in full-term neonates, and you see some kind of correlation, even if there are, of course, some noise. Then in a second time, we tried in the second step, we tried to evaluate the directionality of this coupling. And for this, we actually wondered whether it's microstructural changes that are driving functional changes or the reverse or parallel mechanisms that are interacting with each other during development. So to assess this question, she actually analyzed data from preterm babies that were scanned close to birth and at term equivalent age, so longitudinal MRI, and she evaluated whether the changes in microstructural connectivity or functional connectivity are related with the reverse marker very early on in the first session or later on in the last session. Unfortunately, the relationship were all present, but none were super different from the others. So for now, we couldn't conclude from this and really say that one is more driving the others or the reverse. So I think it's an interesting way to think about our markers in MRI and try to understand whether the microstructure is driving the functional specialization or the reverse, but still some work needs to be done, potentially in larger cohorts or at different ages, to investigate this further. Another studies that I would like to mention is the ones that we've done to relate MRI markers and EEG functional markers. So for this, we actually focused on the visual system because it's a quite simple system and it can be assessed quite easily in babies with evoked potentials. And you might know that when you do visual stimulation, one of the first potential, which is called the P1, has this strong peak here in occipital regions. And when you actually look at the latency of this peak, you see a strong decrease with age. So at birth, you have a latency that is around 250 milliseconds, and this latency is decreasing in a few weeks, in 15 weeks or so after birth, to around 120 milliseconds. And what has been supposed for years is that it's the myelination process of the white matter fibers that is accelerating the conduction speed, and that leads to this decrease in latency. So we actually tested this in a group of babies. We actually observed an increase in the speed. And what was interesting is that for these very same babies, we actually had some diffusion MRI, so we could evaluate the myelination of different fascicles. And what we observed is that this speed is strongly linked to the diffusivity metrics in the optic pathways, in the optic radiations, meaning that when the pathways are more mature, the speed is higher. And of course, for this, we took into account the fact that there were some age differences across the babies too, so that it was not a factor influencing both markers. More recently, we actually did the same experiment, but with more complex stimulation. So it was not central stimulation, but lateral stimulation. So when you stimulate only one hemifield, you first have a contralateral response, and then you have a transfer of the information to the ipsilateral hemisphere. So we tried to relate the speed of the contralateral response to the optic radiations, and we observed similar correlation compared to the very first study. And when we looked at the transfer of response from contralateral to ipsilateral hemisphere, we also observed some correlation with the maturation of one specific bundle, that was the fibers of the corpus callosum connecting the occipital regions. So these two studies actually show that the efficiency of the brain responses to visual stimulation are strongly dependent on the maturation of the connections. In a later study, actually, we tried to do the same for the auditory system, and actually the relationships were much less clear, much more complex, possibly because the auditory system has maturation that starts before birth and lasts for a longer period compared to the visual system. So potentially the mechanism at play for the auditory system are more complex. But one interesting aspect that we also tried to look at is for the auditory and more precisely for the language network, is to which extent the functional lateralization that is developing in babies might be related to anatomical markers. So you might know that there is a strong lateralization in adults in favor of the left hemisphere, and this is actually also the case in babies. So there have been several works by the team of Gislander and Lamberts to assess this. And we wondered whether these functional asymmetries might be related to anatomical asymmetries. So we actually looked at asymmetries in brain folding and observed some asymmetries very early on, mostly in perisylvian regions, which are at play for the language networks. And we also observe strong asymmetries of this very little fascicle, the arcuate fasciculus, that is actually connecting the parietal, temporal, but also frontal regions to make them communicate to process language. We also observed some maturation differences in these very same regions in infants. So all this suggests that potentially very early on there is a bias towards the development of one hemisphere compared to the other. That will be the basis for developing functional lateralization. Even, of course, we can't really say what is the starting point, or whether maybe it's functional lateralization that starts first in one hemisphere and leading to some bias also for the anatomy. Another aspect regarding the language network is the way the bundles are maturing, developing. So for the case of this, we actually dissociate different bundles, pathways for the language network. On one side, the dorsal pathway with this arcuate fasciculus that I mentioned before, compared to the ventral pathway with the uncinate middle longitudinal fasciculus, but also the inferior frontal and inferior longitudinal fascicles. So in this study in babies, we actually observed that there is an earlier maturation of the ventral pathways, and that the dorsal pathways are maturing a bit later, but with a catching up during the first weeks after birth. And this was actually quite coherent with what has been described in non-human primates, because in this non-human primates, it's mostly the ventral pathway that is developed. And the catching up of the dorsal pathway, we suppose that this was related to the development of cross-modal representation of speech in infancy, because when the baby is trying to make sense of his environment, he is hearing some sounds, but also looking at faces, speaking to him, and trying to imitate, so producing, involving the motor regions to produce some sounds that look like the ones that he is hearing and seeing. Maybe one last point before I move to the last section is about the fact that also at the functional level, so I mentioned that there is this asymmetry of this lateralization in the activations for language, but there is also some asymmetries in the latencies that we can measure. So this can be assessed with EEG when you do monaural stimulation, meaning that the baby hears only one stimuli at a time in one ear. When you do this, for the auditory system, actually you have responses both in the contralateral and in the ipsilateral hemisphere. And the latencies are actually not exactly the same, depending on whether you stimulated the left or the right ear. Here you have the responses on the left side to stimulation of the left ear that is longer than all the others latencies. And you don't see this when you look at babies without corpus callosum fibers. So we interpreted this thinking that this longer latency is due to a transfer of response from one hemisphere to the other. And again, we tried to relate the speed of this specific response to metrics obtained with diffusion MRI to assess the maturation of callosal fibers, and we observed some kind of correlation. So this suggests that potentially very early on you might have asymmetries, but also asymmetric transfer of information from one hemisphere to the other through the callosal fibers, and this might make the further development of lateralization in the case of the language network. So I hope this was more or less clear, and now I would like to give you some short examples on how we can do some studies also to have a look at the impact of prematurity on the development of the brain. So you might know that children born preterm are at higher risk of neurodevelopmental disorders than the ones that are born at term. And here you have a plot coming from the EPIPAGE-2 study, which is a French study looking at the outcome of children. They have evaluated them at 5 years of age. And here the plots actually shows the survival as a function of the gestational age. So let's remember that full-term birth is 41 weeks of gestational age in France. And here in particular, in pink, you have the survival without any neurodevelopmental disabilities. So you might see that it's only at late ages that you reach a threshold of 50%, meaning that more than 50% of the babies born preterm will show during childhood at least some neurodevelopmental disabilities, even if these are not severe or moderate. These can still have some impact on their whole life development with some school disabilities, for example. So for these children born preterm, we actually need early diagnostic markers to better understand the brain abnormalities, to better quantify the dysmaturation, and also prognostic markers to know how this will relate to future behavioral deficits and disorders. And this is super important to set up very early on some interventions to improve the outcome. And we need these interventions to take place early on to benefit from the intense brain plasticity during this early postnatal period. So to look at the effect of prematurity, we've done a few studies. Some of them have looked at the folding process that I mentioned before. So we actually, when you look at preterm babies without brain lesions, we haven't seen big differences in terms of brain volume, but still there are some more subtle differences in terms of folding. And in particular, recently we were interested in one specific fold, which is the central sulcus that delineates the sensory and the motor cortices. And this sulcus is known to show representations of the body. In particular, you probably know about the end knob. So this part of the sulcus showing a knob, and that is the place of the end representation. So the team of Jeff Mangin have developed tools to evaluate some characteristics of shapes of the sulcus, and we used this to look at the folding patterns in preterms. These preterms were assessed very early on, so at around 7 months of pregnancy, and one time later at term equivalent age. So we analyzed their folding patterns early on, and we evaluated to which extent these folding patterns might predict the outcome of the preterms that were assessed a long time after, at 5 years of age. And what we observed is actually not super clear prediction, but at least some not null prediction for handedness and also for fine motor skills. So there was a quite relevant classification of the preterms. For example, here you have all the sulci. In red, the preterms who show a poor outcome in terms of fine motor skills at 5 years. And I hope you see that these red sulci are positioned differently compared to the gray ones, suggesting that their shapes is actually quite different. So this is one of the examples of using early markers to try to know how whether the kid will have some disabilities later on. We have tried to do the same based on markers of cortical microstructure. So these are, again, some work from Andrea Gondova. So I forgot to mention that before the folding stuff were the work of Héloïse Desvareilles, another bright PhD student. So with Andrea here, we actually looked at the cortical microstructure. First, to do a very simple task of predicting the prematurity status, meaning whether the infant is born preterm, yes or no. And so she developed machine learning approaches to get quite good classification between the two groups of babies. And actually, she showed that there are different clusters of regions of the brain that contains relevant information to perform this classification. In a second step, she looked not at scans obtained at term equivalent age, but at scans obtained close to birth. And again, the classification was quite good, but this was highly dependent on the size of the cohort that was used, meaning that if the cohort was too small, the prediction was not different from the random level. But actually, in a third step, she looked at whether this microstructure contains more relevant information for the prediction of the outcome of the babies. And unfortunately, this was not the case when she looked at different markers of functional neurodevelopment. For example, here you have the scores for the motor outcome. And here you have the plots showing the prediction capacity that are actually not different from random level. So this suggests that predicting outcome from the microstructure is not so easy. And this was actually initially done in the scope with the objective to replicate some findings by others, and we were not able to replicate this. Beside cortical microstructure, I've mentioned before the maturation of white matter. So we've done also some studies, in particular to look at the sensory motor network, dissociating different tracts, connecting cortical regions and subcortical structures. And here's the interesting thing that we observed some vulnerability patterns that are actually different across the connections, with the cortical-cortical connections, for example, being more impacted by prematurity than the subcortical connections. And even between the cortical and the subcortical connections, we observe some differences. These vulnerability patterns of the connections were mostly following the maturation, the typical maturation of the connections, meaning that because prematurity probably is occurring at a time when these connections are maturing intensively. Again, we looked at the association with outcome. We observed some associations at the group level, but when we used some prediction methods to get some to go to these prognostic markers that I mentioned before at the individual level, our group was not big enough, and there was too much variability to obtain some good things. Last thing that I would like to mention before concluding is a big study that we are trying to develop in the hospital where I work in Paris, the Hôpital Robert Debré. So we are actually following preterms in close collaboration with the neonatal unit, because preterms have MRI scan for clinical purposes. And we took part of a study trying to evaluate the impact of a locomotor training with the team of Marianne Barburot. And for this project, actually, the babies had some training at home with the parents for 2 months once they was discharged from the hospital, and they have a very precise neurodevelopmental evaluations to see whether the training improves their development. And in this context, we actually wondered whether we can get some more precise understanding at the brain level of the plasticity mechanisms that are at play with this training. So we added some MRI, we added also some EEG for research to understand the development of these preterm babies without training, but also, of course, to look at the impact of training. And now we are actually following these kids at 3.5 years of age. So we are still in the process of analyzing all the data, but hopefully in the next 10 years, we will have some results about this. So just to conclude, I hope that this talk has shown you that the infant brain shows an early architecture in networks, but that the functions rely on networks that are maturing at different rates and with calendars. So we need longitudinal multimodal study to really characterize these individual trajectories of development, because the variability is quite important across individuals. And these trajectories are also important to understand the impact of early disturbances and risk factors of neurodevelopmental disorders. And this is important, finally, to predict outcome, implement early specific interventions, and finally, hopefully, improve the quality of life of many children. So I thank you for your attention, and I thank the parents, the babies that take part to our studies, and of course, all my colleagues involved in this research. Thank you.
Thank you very much, Jessica. It was very impressive. I got like really fascinated by all the research you presented and how you link it up with neuroanatomy and the former work from Pascal Rakik. Before I ask my questions, I will ask if anybody on the board of people that are connected have questions, or if there is any question online for Jessica.
I'll check YouTube. Hold on.
All right. So while you check YouTube, I'll ask my first question. So in your introduction and what you're showing, you suggest that the synaptic pruning in human babies is happening, I think if I remember well, your chart after 6 months, something along that timeline. But if I remember my reading of Pascal Rakik paper that was done in monkeys, it seems that like the synaptic pruning was happening right after birth. And so you think like there is a difference between, there's obviously a difference between men and monkeys, but in the degree of maturation, the brain has when the baby is born between those two different species, what would it be the case? What would we be born with an, not as well maturated brain? Do you have any suggestions?
So that's actually, my hypothesis is that actually it's definitely the case, because it's the first plot that I showed you with these strong changes in development, this strong phase of growth in the humans that occur before and after birth. And this doesn't seem to be the case in non-human primates. So the role of environment can be higher in humans. So there are actually some discussions about saying that finally the human species is potentially both immature in terms of brain, so that environment can be really important on the development, but also some other, it also shows some mature aspects. For example, we are quite good to process sensory stimuli. And my hypothesis is that it came with evolution, and that being not so mature at birth is an advantage to learn many things from the other environments, such as language, etc. Regarding the synaptogenesis, actually the synaptogenesis is one aspect that closes the plasticity in some way, and the fact that it comes later in humans and than in non-human primates goes in this direction. So does it answer your questions?
Yeah,
and
it does.
regarding the plots that I've shown, so it's plots done in humans by Eton Losher and Dalokar, and they say when you look at the peak of synaptogenesis, it's around 15 months for the visual cortices and more around 65 months for prefrontal. This includes the 9 months of pregnancy. So we are close to the 6-7 months that you were mentioning for the visual cortices, but for prefrontal,
it's much later.
Yeah.
Okay. It's interesting because when you have kids, you see those kind of development and questions in such a different way. You're like, oh wow, I just snapped my fingers and you lost 50,000 connections.
Yeah.
Anyway.
Yeah.
Any other questions? I have a list of questions there, so I can carry on. So you should like also those very exciting studies where you demonstrate that there is a correlation, direct correlation with track-specific myelination of optic radiation and corpus callosum, and the latency for P1 in ipsilateral and contralateral. But so, and I'm really terrible at electrophysiology and EEG. So my question is very simple. If I remember well, P1 is about detect like novelty, and was supposedly located in the frontal lobe. Do you think other tracks could be contributing to this P1? I mean, did you explore other connections? Because you went fast through it, so it's difficult to see which connection were explored besides the one that was significant.
Yeah, actually, those P1s that we measured following visual stimulation were really located in occipital regions, so where the frontal regions are. This P1 is probably not the very first response that we could have with EEG, but it was the most robust. So that's why we looked at this one, and also because previous studies showed this really strong decrease in the latency. In the very first study that we did, we actually looked at many different pathways. So we could have we had the optic radiation. It was easy, but we also looked at control tracks, corticospinal. So I don't remember because it was during my PhD, but we looked at several ones, and the optic radiations were the ones coming up in the correlation. So it seemed to be specific. But I agree that maybe another way to assess this would be to use more whole brain analysis without hypothesis on the tracts involved, and maybe this will highlight some other regions involved in P1.
I was just curious. That's great. It's it's significant and it correlates, and you can't help but wonder if other part of the brain contributed to that as well.
So for the other main tracts, it was not the case, but we didn't look at all the white matter. So,
and maybe like a different period of time or age of those little babies will influence also the part of the brain that correlates.
Yeah, for sure. So that was one of the hypotheses we had when we couldn't see on the same period of development, we couldn't see such relationships for the auditory responses compared to different tracts. And we thought, okay, it's because we are not at the same period of development for this tract, for these responses. Maybe we would need to have measures before and also after, because the maturation of the auditory network is much more protracted. It takes longer than the visual system. So yes, I think that for the visual system, it was the ideal situation. Strong maturation, strong development over a few weeks, very strong functional changes, and very strong myelination. And
yeah, it's easier to explain than the corticospinal tract. Okay, I had another question. Like the third part of your presentation, you very nicely demonstrated that there is an inhomogeneous vulnerability pattern in premature children, babies, sorry. And before you made a link between the brain maturation and brain evolution that follows the Evo-Devo hypothesis of the brain, I was wondering whether this vulnerability pattern in any way could be related with evolutionary pattern, whether this is like the latest evolutionary regions that are like the most affected or the reverse way. Why would it be the case?
So as you said, we have this some insights on the fact that with Evo-Devo framework, let's say, the most recent regions are the ones that develop the latest in babies. And depending on when the impairment is happening, according to the calendar of maturation, it might be impaired less or more or less. Saying in other words, these later developing regions and networks, the most recent in the human species, actually they took their basis of development also on other networks. And even if you have disturbances occurring earlier than the development of these networks, it might impair more, let's say, all the networks with impacts on most recent networks. But this is not a super clear response, but I think it's quite complex to evaluate.
Make sense. That does make sense. It's very mechanistic. But you don't think that, like, it's just crazy talk, but you don't think that the region that are the latest evolved on the phylogenetic tree might be the most fragile to pathology, for example, as if, you know, they were not optimized, or I don't know. It's just an idea.
I wouldn't say that. I would say that the networks are differently vulnerable depending on whether they are already developed, yes or no.
I see.
And we say in general that, okay, we have a lot of plasticity, etc., but there is also very early on during development a high vulnerability. So the networks can't be built in some cases. So strong plasticity, but also strong vulnerability. And for sure, the impact of disturbances won't be the same at different times of development of the networks. But I wouldn't, so that's why I think that there will be different vulnerability patterns depending on this evolutionary, how to say, recent networks, etc., but not because they are more recent in themselves, so maybe less robust, etc., but just before they are not developed at the same time, at least for development. I don't know later in adulthood, but
that's a question that I had for a long time. Never found a way to really explore it. So you were at the right place, right time to ask you the question. I have a last question, and you should like a poor association between the brain features measured in baby preterm and the functional outcome that give a lot of hope for plasticity and the possibility for those preterm babies to have a normal life. But how do you explain this variability, because it's still a lot of them actually show some pathology later in life. So what is the secret recipe for these preterm babies that makes them succeed and have a normal life versus maybe having a poorer functional outcome? Because apparently it's not the brain feature that we can measure with MRI.
So several responses to this. Maybe the first one is just to say that it's not because in particular studies we didn't find good prediction that we use the good markers, etc. So I think there are still a lot to do to improve what we can do to have these early markers with MRI, but also with other explorations, combining them, EEG, clinical information, etc. So this is a super important aspect. We are not there yet, but hopefully in a few years we will be there. The other aspect is that, yes, the brain still is important because in some cases it's clear that there will be some impairments. What we need is to be able to predict even in the cases where it's not super clear. So you don't have a big lesion, you don't see big abnormalities, but you know that in other groups, this kids are more at risk to develop this. So the brain is important, but it's not the only thing. And when you look at actually in preterms, what are the factors influencing development? One of them for cognitive development is definitely the family, the socioeconomic status of the family. So even two babies with the same brains at birth won't develop in the same way. So we know this in typical babies, and it's also the case in babies at risk of neurodevelopmental disorders. So I would say for these babies, having interventions that are even not really focused, but that aim to really stimulate language if it's not done sufficiently at home, or stimulate the connection, cognition, etc. This can be important in any case.
I see. It's fascinating. It's fascinating.
Sorry.
Go ahead.
No, linked to that, I was wondering whether you have so first of all, sorry, thank you for your wonderful talk. Right. I haven't thanked you yet. That was super interesting. I was wondering, linked to that, whether some striking cultural differences come to your mind, or whether you know of like some cultural differences that occur and you are visible in the brain data of babies. Do you know of any that could, we can trace back to culture? So because you economic status, you mentioned all these things that can be measured across cultures, of course, but do you know of any? Yeah.
So one first response, but maybe not really a good one, but I mentioned at some point that initially the babies have strong discrimination capacities of the phonemes, and this is the case in all neonates, discriminating phonemes from all languages, and you lose this capacity when your brain is not exposed to some sounds of the language. So you become actually in the first year an expert of your culture through the language in some way. There are also strong cultural differences, for example, in the way to take the babies, to carry them, to let them by themselves, and this make also a big difference at the motor level. We haven't looked, I don't know about studies that have looked at brain differences of this, but behaviorally, you see strong differences in the way the babies develop their posture, their motricity, depending on whether in their culture, the mom carry them all the time, yes or no, etc. This makes a difference. But I think that would be super interesting to look at brain markers of these behavioral differences that we see during the first year, actually. So initially, same brain, same babies, but some divergence.
It's interesting you're saying that they become experts of their culture in the first, at some point, the more exposed they are to it. That links basically to what you're saying. We're or babies, when babies come to world, they have on the one hand a very developed brain because they have the capacity to become an expert in every culture in that sense, or speak every language, right? And then they learn something, but it's actually decreasing as well at the same
time. That's it. They have a brain already organized, able to process many things, but not fixated, with the capacity to evolve with the environment. And by becoming experts, they lose some capacities, yes. That's my view.
Funny, there's this one study that is studying children in Zambia, and in one of the languages, there are many languages spoken in Zambia, and one of the languages, they don't have the terms for left and right because the orientation is not egocentric as we have it, but allocentric. So they're orienting themselves based on the environment. And the more English the kids learn, because they learn English in school, the better they become in English and learn the terms for what is left, what is right, the more they become, or the shift goes from their allocentric to an egocentric orientation. It's a similar concept, because they basically, you learn something, you learn English, you actually learn a language, but then at the same time you lose a capacity to it affects something completely different as the orientation.
And this also shows that all the functions are actually developing in connection with each other, and it's you can't just say, okay, let's study one thing at a time, because in real life, in babies, it's not the case, and you can't manipulate the environment to really dissociate the different factors.
That's right. Thank you.
All right. Another question, Eva? Go ahead.
Thank you for your talk. It was really interesting. I learned a lot. I was wondering, since we're talking about the importance of the environment in shaping and correcting, maybe if there has been some damage, modulating this pruning. I was wondering if you think with the environmental stimuli, we can do as much as we would need to help the kids, or there is a place for some other kind of stimulation, like brain stimulation in some cases that would, or is there something that we cannot do by environmental stimulation that there would leave a space for another?
I would say that we have not explored all the possibilities of stimulation in babies, also environmental stimulation. So I mentioned this locomotor training, which is quite original, because initially people wouldn't have thought that we could stimulate the locomotion of a baby very early on. But my colleague Marianne Barburot had this idea that yes, it should be possible, and I think it could be the same for other modalities. So I think there are still places for many ecological stimulation early on to improve outcome of babies. People have focused a lot in the clinics with messages or carrying the babies, but there are other fields of research with music, etc. Besides this, all the things with brain stimulation, etc. I think for babies it will be more difficult to get the ethical authorization for this, at least for now. Maybe in a few years, people will have changed their mind in the way that on how to care with the babies. But for now, I'm not aware of studies with TMS.
I guess the ethical consideration would come if there is a space for...
But it might come, because there are now some studies with stem cells, for example, with preterm babies to restore brain tissues that have been damaged very early on. A few years ago, I think it wouldn't have been possible. So now it's done. It seems to work very well. So there are a European consortium with, for example, the team of Manon Benders working on this. So yeah, I think there are still many possibilities to explore in the next years.
Thank you.
Any other questions?
I see. I'm in the dark now.
Yeah, that was how late is it? I don't think there is any other question. So thank you very much, Jessica. It was fascinating. It was an excellent talk, and I really captured my curiosity. So I'll follow up asking you more questions in the future by email.
Sure. Thanks a lot.
Thank you, Jessica.
Very much.
Bye.
Bye.
Bye-bye.
That was a very good talk.
We are still live.
That was okay. That was a very good talk. Still there.
Okay.